Okay, so hello everyone. Welcome to our today's Zoom seminar. It's really my pleasure to welcome our today's speaker, Dr. John Manion from Pittsburgh. John is actually currently a junior faculty as assistant professor in microbiology and molecular genetics in University of Pittsburgh School of Medicine. So John actually completed his undergraduate study in King's College London, UK, and then continue to finish up his PhD training in University of Sydney in Australia. And then he decided to move again to pursue his postdoc training in Boston Children's Hospital, Harvard Medical School, in microbiology and neuroscience. From 2024, John actually started his independent research program in Pittsburgh School of Medicine. Actually, during his postdoc, he actually did really fascinating work on the bacterial toxin to modulate the immune neuron crosstalk. I'm actually read his paper. It's really, really fascinating. The main goal of John's lab is to understand the machinery by which the bacterial toxin how they cause the disease, and then through discovery of their cellular and molecular targets. So as we know that a lot of pathogenic bacterial secreted large protein, which targets cells and then enter the cells and deliver enzyme which can potentially damage or rewild the cells. These called bacterial toxins. So John's lab, as I just mentioned, it seeks to understand the machinery of how these toxin works. His lab mainly interested in understanding how bacterial toxin play a beneficial role for the bacterial in processes such as competition and colonization. And also his lab using these bacterial and toxins as tools to understand fundamental physiology. Because the demands of these toxins, they target sites critical to a physiological function to achieve their goals. One example is the TCD-B, large toxin produced by Clostridia difficile, which a major cause of serious diarrhea after antibiotics. So TCD-B selectively targets sensory neurons and then pericytes within the intestines through cellular receptors. Therefore, toxin can function as a lens to understand the physiological roles that interactions between the cells and play in normal condition and disease function within the intestine. So through the study of these toxins, John's lab aimed to explore many aspects of physiology and build tools that allow us to hijack cellular physiology. So as I mentioned that John really did a fantastic work during his postdoc training to illustrate these toxins, how they actually play roles during the neuroimmune crosstalk, which I think he's probably going to tell us a bit today. But without further ado, John, thank you so much being with us today, and we are very much looking forward to your talk.
Cool. A real pleasure to be here. Thank you so much for the invitation as well. And yeah, as you were just saying, I'm going to be discussing today mainly work conducting in my mentor's lab, so Dr. Min Dong at Boston Children's Hospital, a fantastic mentor and a fantastic place to do science for anyone who's also considering postdocs and things like that. But today I'm really going to focus on sort of this understanding of how toxins target particular cells within tissue to sort of trigger their physiological functions. And as Tran was just saying, for C. difficile, our findings really led to the discovery that pericytes and neurons within the intestine were really critical targets of these toxins through their selective expression of cellular receptors. Now, hold on. Now, I think like this sort of overall topic is actually very old, this idea that there are these interspecies conflicts. And I kind of like to sort of start out by sort of giving a little bit of the history of immunology and microbiology and how they kind of influence how we think about some of these topics today. So, I mean, as many of you know, actually toxin biology is sort of the home of both immunology and microbiology. One of the greatest discoveries really was these early identification of these potent bacterial toxins, so things like diphtheria toxin by Yersin and Roux. And this is actually what led to sort of the fundamental basis of immunology, with the first serum th therapy coming shortly after in about 1901, obviously from von Behring and Shibasi. At much the same time, Paul Guerre was doing these really interesting sort of growth assays where he was defining that different species actually produce molecules that could inhibit other species in their growth patterns. And this is work that really kind of influenced Alexander Fleming when he was sort of discovering these antibiotics, right? And at the same time, Howard Florey, working more on the physiological side, he actually discovered that nerves secreted mucus, and these were critical sources of these lysozymes, so these bacteriolytics. So the nerves were stimulating these bacteriolytic molecules. And that's what really gave him the insight to actually look further into the work of Fleming and subsequently develop some of the most important therapeutics that we use today. So these antibiotics. And of course, those structures were then solved by Dorothy Hodgkin in beautiful work back in the 60s. And sort of this like idea that there are these different toxins targeting different organisms is obviously not new. But one of the sort of interesting things for me as sort of a neuroscientist is that many of these toxins actually exploit attacks on peripheral nerves. So many different toxins from many different organisms sort of coalesce around this as a target. So one example is like these snakes, they produce these neuromuscular toxins that can inactivate prey for predation. Or, for example, plants, they produce toxins that can act on sort of sensory neurons that sense pain, to cause pain to prevent you from eating the plant. Similarly, because I'm an Australian and every Australian has to study at least one deadly venomous critter. Actually, during my PhD, I studied the molecular mechanisms of box jellyfish venom, and this really drove my interest in understanding how toxins could sort of influence the nervous system and cause effects on the host. So sort of the most potent of all toxins are these bacterial toxins, because they're very large and they have these really selective mechanisms. So they all need to achieve pretty much the same thing. They need to bind to a cellular surface, internalize into the cell, and then they deliver some kind of toxic cargo that inactivates that cell. What's really interesting about this is like every step of this process is really specific. So they bind, in the case of botulinum neurotoxins, to receptors that are selectively expressed on synaptic vesicles. They then extravasate through those synaptic vesicles and they cleave the snare proteins that enable synaptic vesicles to be released. So the molecular specificity of this is very high. And that's how something like a bacterial toxin can achieve that level of potency. So this really influenced me actually joining Min's lab. I mean, his really sort of claim to fame and one of his greatest contributions is really discovering the receptors for these botulinum neurotoxins. So he discovered the receptors for both botulinum neurotoxin A and B, and discovered the very specific process by which that these intoxicated the cells. And as I joined his lab, what I really wanted to understand is what is the cellular logic that underlies some of these other really potent large bacterial toxins? How do those molecules cause their effects in physiology that are very specific to those bacteria rather than other bacteria? I think a lot of interest has been in sort of understanding how some of these toxins cause things like cell death, but at much lower concentrations, they have really profound effects on physiology, that are really the major pathophysiological mechanisms of these toxins. So I was particularly interested in C. difficile colitis. This is a really interesting bug. It's not only really therapeutically important, so there's about 200,000 hospitalizations caused by this bug every year in the US alone. But it's also a fascinating bug from sort of the history perspective. When it was first isolated, it's actually isolated from healthy newborn infants. And actually, healthy newborn infants often are colonized with C. difficile, but has no effect on them. So they have toxin, they have the bacteria, but there's no effect on healthy infants. In contrast, if you take antibiotics, you get overgrowth of this bacteria. It causes a really nasty infection that leads to this really profound damage to the epithelium and really this very strong vascular inflammation. So this really profound edematous inflammation, this massive sort of exudate, and a lot of hyperemia, so the reddening that you see there, that's really indicative of a higher level of blood flow and a higher level of vascular leakage. And if you look at some of this pathology taken from the colon, you can look at some of those really typical features. So you can see there's a really strong neutrophilic infiltrate, there's a really strong edematous component, and there's also these quite sort of pathognomonic these eruptions of neutrophils out of the epithelium. This is some of these pseudomembranes here. So as a disease for a toxin biologist, it's quite good because this is an entirely toxin-mediated disease. So the expression of these toxins is really required for the disease. And what we think is sort of the major toxin for this disease is toxin B. And this is one of these really large, multi-domain bacterial toxins. So it has all of these different functional domains that are going to intoxicate the cell. Toxins like toxin B, I really think of them as tiny little nanomachines. Like they're actually extremely complicated because they have all these different functional domains, and their sort of synergy in achieving that intoxication process is very high. So in the case of TCDB, what it actually does is it binds to two distinct cellular receptors in two different sites. So it binds to these frizzled class of receptors through one interaction, and it binds to the CSPG4 class of receptors through completely separate sites on the toxin that are actually completely functionally independent. So these intoxication processes occur in different cells at sort of a different time point. The toxin then undergoes clathrin-mediated endocytosis, and subsequently, acidic conditions within the endosome enable the toxic cargo to be delivered into the cell. In the case of TCDB, this toxic cargo is this glucosyl transferase, and what it's going to actually do is it's going to inactivate these Rho GTPases, and subsequently that causes actin disassembly within the cell. And that leads to some of the really classic interactions you observe with these toxins. So in sort of like really interesting work done both in the US, actually also a lot of the work was also done in China. So they defined the receptors. So CSPG4 was defined as one of the cellular receptors, whereas Frizzled was defined as the other cellular receptors. And the binding sites for all of these toxins have been solved sort of through some of the amazing advances in both CRISPR genome screening and some of these structural studies, which really defined where exactly these receptors bind within the toxins. So I was personally interested in finding out where exactly within the intestine these receptors were most enriched. So which cells could be considered the most sensitive cells within the intestine. Now, really classically, everyone just thinks of these toxins as ones that target the epithelium or targeting immune cells, but no one's really done sort of a no one had really done a sort of investigation from looking where exactly the toxin receptors are expressed and sort of inferring that that might be sort of the pathological origin of this infection. So in terms of in the intestine, when we looked at the expression of these receptors, what we actually found is there's really strong enrichment of these frizzled receptors in the enteric neurons. That's the neurons that live within inside your intestine, their own internal nervous system. Whereas they're sort of not expressed in many of the other typical targets we might be looking at. Um, when we actually stained cultured DRG neurons, what we found is these frizzled receptors are sort of expressed all the way along the axons of the neurons. So they're expressed all the way along the neurons, not just in the cell body. So they're very much available as targets for these toxins. We wanted to sort of try and understand how exactly the toxins' physiological effects could be tied to that expression. So we did some really sort of basic studies to try and understand how the toxins function within tissue. So in this approach, what we do is we actually administer the toxin into the cecum of the mice, basically, and then we collect the mice at different time points. We do pathology and we look at the effect of the toxin on the mouse's intestine. And what we actually found is that if you inject something like a vehicle, it has no obvious effect on the intestine. Whereas when you inject these toxins very rapidly, you observe this kind of edematous inflammation. So that gap here, sorry, probably I hope you can see my cursor. Anyway, basically you observe this gap in the between these layers that gets bigger and bigger and bigger, and that's the sort of edematous inflammation. So this occurs really early in the context of the infection. So, sorry, in the injection. So straight after you ingest this toxin, you already get this kind of swelling response within the intestine. And this actually kind of occurs before much of the epithelial changes. So this is one of the earliest changes that is occurring inside of the intestine. And through my own training, I actually trained in the lab of Steve McMahon when I was at King's. And one of the things Steve had discovered back in sort of the 1980s was that if you actually stimulate the extrinsic neurons leading to some of these hollow organs, you actually cause this edematous inflammation. So you cause this swelling, you cause this vascular leakage phenotype. So this is entirely caused by something like electrical stimulation, whereas if you denervate the tissue, you no longer get this kind of effect. And similarly, in sort of beautiful work done by Harry Pofalakis over the sort of a couple of decades ago, he'd actually found that when you inject toxin B into the intestine, if you actually remove some of the components of neurogenic inflammation, so you get rid of some of the sort of causes of that kind of inflammation, it actually completely protected the epithelium of the mice. So by blocking this kind of neurogenic process, you're actually able to protect the epithelium, which really suggested to us this is a really kind of profound cause of this kind of inflammation that's occurring with this kind of toxin. And indeed, when we look at the neurons that are sort of expressing these toxin receptors, a really sort of common feature of all of them is they express these neuropeptides that are really commonly associated with classical neurogenic inflammation. So one of these neuropeptides is CGRP. It's actually the most potent vasodilator known to man. So if you just inject it into the skin of a human, you very rapidly get this sort of erythema response. So you get this rapid increase of blood flow to that tissue. And similarly, many of them also express substance P. And it's the same story with substance P. If you sort of inject substance P into the vessels of the mice, it will cause generalized leakage all over the tissue. And this is very much caused by these gap junctions being disrupted within sort of minutes of that neuropeptide being injected into sort of an animal. So we wanted to sort of find out if this is also the case in our model. So again, we turn to that cecum injection model. We administer the toxin into sort of these genetically modified mice where we've taken out different components of their sort of neurogenic inflammation apparatus. And what we found is that if you remove substance P, so you remove the gene that makes substance P, or you remove its receptor, you block the ability of that toxin to cause those responses within the tissue. And the same was also the case when we knocked out CGRP, in particular, actually the CGRP that mainly comes from the intrinsic enteric neurons, rather than sort of CGRP that you might more be sort of classically aware of, so CGRP alpha. And indeed, this is also true in the infection model. So in this model, what we basically do is much like humans, if we give mice antibiotics and then gavage spores of C. difficile, they actually become sensitive to the infection. And when we do that, if we knock out these components of neurogenic inflammation, we can dramatically protect mice from this kind of phenotype. So you can protect them from that edematous swelling, and indeed that neutrophilic infiltrate and that damage to the epithelium by blocking these elements of the pathway. We wanted to understand what the molecular mechanism of these toxins was within the tissue. So we turned to an approach where we can basically culture adult sensory neurons from mice. So if you actually take out the dorsal root ganglia of mice, they can be cultured pretty happily. It's a pretty unique neuronal model in that most adult neurons don't really like being cultured. So when you treat these cells with TCDB, they very rapidly cause the secretion of these neuropeptides, in particular substance P, and this occurs at extremely low concentrations. We wanted to show that this was really dependent on the functions of this toxin. So we have these mutant toxins where we've removed their ability to interact with these Rho GTPases and cause that enzymatic effect, and that blocks the ability of this toxin to cause secretion. And similarly, if we remove the ability of that toxin to bind to frizzled, so we introduce mutations in the residues that bind to those frizzled proteins, that also disrupts the ability of these toxins to cause substance P secretion. We then sort of turn to a really classic model of neurogenic inflammation. So if you just inject something that causes vascular leakage into the footpad, you can actually block these with things that can block neurogenic inflammation. So if you inject capsaicin, it activates TRPV1, it causes the secretion of some of these neuropeptides that will cause the swelling of the mouse's foot pad. If you inject TCDB, it's very much the same. And if you remove the ability to interact with those Rho GTPases, you also remove the ability of C. diff toxin to do this. You can block this with antagonists that block substance P receptor activity. And similarly, you can also block this function by blocking the ability of that toxin to interact with those frizzled proteins, whereas this doesn't actually is not actually impacted by the ability to interact with the other receptor interface, so that CSPG4 binding interface. Inside the intestine, we can also do similar assays. So we can inject the toxins into the intestine and then actually measure their secretion of some of these neuropeptides. And again, very rapidly after you inject these toxins, you already start to see secretion of these neuropeptides very early in the onset of the disease, and also very much correlated with sort of the degree of edematous inflammation. And the ability to cause that secretion is very much attenuated by removing the ability of that toxin to interact with those frizzled receptors. So in summary for this part of the talk, I hope what we've sort of explained is that the C. difficile infection itself relies on these toxins, that there's a neurogenic component to this kind of infection, and that component is really dependent on the secretion of these neuropeptides. As a molecule, TCDB acts as a pro-secretory molecule in neurons, and it subsequently causes CGRP and substance P secretion, and that this interaction depends on the neuron's expression of those frizzled receptors. So this leads us to the obvious question. So if the major sort of target of those receptors is the neurons, what's the point of also binding to CSPG4? Why do they need this whole different binding apparatus to bind to a different cell within tissue? And what could be the cellular target for CSPG4? And there have been sort of a number of different studies that have claimed a number of different kind of targets for CSPG4. So we really wanted to nail down exactly where in the intestine this was really expressed. And we have these really beautiful labeled mice where we basically have a CSPG4 DS-Red mice. They're available from JAX. They're really easy to kind of image. So we image those mice. And we sort of co-stained the endothelia because we realized that actually in the intestine, the expression of the CSPG4 was exclusively in these cells that wrap the vasculature. So they beautifully wrap the vasculature here. And actually, all of the CSPG4 expressing cells within the intestine are these pericytes that wrap the vasculature. Now, these go right up to the mucosal surface. So just beneath the epithelium, you have these pericytes wrapping those endothelial cells, and that's just sitting just beneath the epithelium, just in the right place to kind of be intoxicated by these toxins. So we sort of did some staining studies to see if these were expressed in any other kinds of cell, and we found yes, they're basically coalescing with the CD31 positive endothelial cell. They're not expressed at all in sort of lymphatics, so there's no lymphatic and like perilymphatic expression of this receptor. Similarly, there's zero expression in enteric glia, so those are other cells associated with this kind of complex, but they don't express this, and indeed they're not expressed in other classes of fibroblasts. So classic fibroblast markers like PDGFRA, they won't express it. We then turned to a cultured model of pericytes. We took actually human brain pericytes and we treated them with TCDB. And we actually found that brain pericytes were one of the most sensitive cells that we had tested for this toxin. So a really classic effect of these toxins when they cause that actin disassembly is they actually cause this cytopathic cell rounding effect. And when you treat cultured pericytes with TCDB, they cause this at a really low concentration. So the CR50 might be sort of about 10 to 15 femtomolar. So it's a tiny amount of toxin. And when you've removed the ability of that toxin to interact with CSPG4, it decreases the ability to cause that cell rounding effect. And this is not really influenced by the ability of that toxin to interact with the frizzled receptors. Similarly, we actually did a multiplex assay, and we found that the pretty much the one cytokine that is induced by TCDB on pericytes is actually IL-8, so CXCL1 in mice, and they cause the secretion of this cytokine. And again, this was dependent on their ability to interact with CSPG4. Interestingly, again, this occurs at really low concentrations, so about 60 femtomole is sufficient to cause that secretion. We then conducted the same kind of studies in vivo. So we found that in mice, indeed, TCB causes really strong secretion of CXCL1. This has been really well established. In fact, mutations in CXCL1 actually cause enhanced sort of C. difficile. So if you have mutations that cause more CXCL1, you're going to have worse C. diff. And indeed, in our mice, like it causes really strong secretion of CXCL1 that's pretty much entirely abrogated by removing the ability of that toxin to interact with CSPG4. And this was also phenocopied in mice where we knocked out the CSPG4 receptor completely. And indeed, in those mice, there's no longer a sort of a benefit to removing the CSPG4 binding ability. So we then had these two sort of different interfaces, and we were quite interested because neurons and the vasculature have this really long sort of co-history. Like neurons are well known to follow along the vasculature. And as we were saying, many of these neuropeptides, they're actually kind of vascular neuropeptides, right? They're causing things like edema. They're causing things like swelling when they're treated to tissue. So we were really interested in seeing what was sort of the cellular architecture that was occurring between these different toxin sites. And this has a super long history in the intestine. So some of the early drawings were actually done by Santiago Ramani Cajal. And what he found was that basically the intestinal neurons followed along the vasculature and we found pretty much the same thing when we looked with sort of more modern techniques. So here looking at those pericytes, and again, the neurons sort of coalesce along the vasculature. And in the intestine, when we sort of imaged some of these particular classes of neurons, we found it formed extremely tight appositions with the neurons, which look like almost synaptic-like appositions, particularly at the branch points of these pericytes, where these pericytes kind of locate at branching areas of vessels. So here you have like this kind of triskelion type complex where you have the vessel running through three different ways. And at those points, you actually find neurons. So they're a really kind of interesting physiological structure kind of revealed to us by one of these toxins. Um, so indeed, those pericytes are innervated, particularly by these CGRP producing neurons. So these CGRP producing neurons act to those particular sites. And indeed, if you treat cultured pericytes with really low concentrations of CGRP, they actually cause IL-8 secretion already. So they're able to act on these pericytes and trigger some of these immune functions very directly. And this sort of led us to a kind of interesting aside. Perhaps many of sort of the kind of neuroimmune phenotypes are mediated by this kind of really direct interaction where the neurons can act on things like the vasculature to actually control immune cells very indirectly, subsequently leading to many different and very confusing phenotypes. And indeed, when we looked back into the sort of historic literature, Howard Florey as an Australian is obviously a big influence. And he had previously observed indeed these pericytes within the intestine. And more interestingly, when he actually looked at some of his photos, we actually observed that there are indeed nerve terminals contacting these pericytes within the vasculature. So that had been observed as early as the 60s. So then sort of going on from that work, what we really wanted to understand is which exact neuronal subtypes within the intestine could really contribute to this kind of inflammation that we had been observing. Now, the GI tract, I think, needs sort of no introduction. It's like one of the most complicated nervous systems that I know. So I was really grateful to have the collaboration of Meenakshi Rao, a really talented and fantastic pediatric gastroenterologist. And the nervous system in the intestine, I like to break it up a bit more simply than maybe some of the proper neurobiologists. So I like to think of sort of about the extrinsic and the intrinsic influences. So the neurons within the intestine versus the neurons from outside your intestine. And we can kind of luckily sort of simplify some of this because most of the neuropeptides were expressed in really particular subsets of cells. So we were mainly focused on the intrinsic primary afferent, so the sensory neurons of the intestine, and the extrinsic primary afferent, so sensory neurons that come from outside the intestine. And we really wanted to understand which of those cells might be contributing to this phenotype. Now, there are a lot of different ways to sort of understand how different cell types contribute to phenotypes within tissue, and they all rely on pretty much the same principles. So you express some kind of exogenous receptor, and then you control that receptor with different influences. So something like chemogenetics, right? We're expressing an exogenous receptor that's responsive to a chemical, and in optogenetics, we're expressing an exogenous receptor that's sensitive to a light. As a toxin biologist, we really like using toxins. So this really classic system, basically you express diphtheria toxin receptor because mice are naturally insensitive to diphtheria toxin receptor. You can make any cell you want basically sensitive to diphtheria toxin. So you've made that cell sensitive to diphtheria toxin. Diphtheria toxin itself is kind of a little bit boring. What it does when it inside tissue, it just kills the cell. So it's a really potent cytotoxin that kills the cell with really high efficiency. We were interested in kind of extending this method. So basically using this as a method to study other toxins and their toxic mechanisms by attaching different toxin domains onto diphtheria toxin. Now, diphtheria toxin itself is kind of a well-established delivery mechanism. So lots of people have been delivering things into cells for years. But one of the sort of opportunities enabled by sort of combining some of these systems together is it gives you a really nice way to understand how different toxin intoxication events can function within tissue. So I like to call this system a toxogenetic system as opposed to chemo or optogenetics. And in this system, basically what we do is we take the enzyme part of the C. diff toxin and then we use inactive diphtheria toxin to shuttle that into particular cells. So in the case of our project, we basically wanted to shuttle this into different cells and understand how they contributed to the pathology. So basically what we have to do is express this receptor in the cells of interest, and then that will deliver our GTD domain into the cell of interest, subsequently intoxicating just that cell. One of the really cool advantages of sort of modern technologies, we have a really precise ways to express receptors in really precise cellular subsets. So these really nice engineered AAVs via the Gradinari lab, they enable us to get really specific expression into neurons as opposed to other cells. So some of you may know TAC1, substance P. It's expressed in lots of different cells within the intestine. So we were really interested in showing whether it was specifically neurons that are involved in that response. So we took a capsid that's really specific for the PNS as opposed to other cells. We infected these TAC1 Cree mice, and this generates mice which have really specific expression only in the neurons inside the intestine that are TAC1ergic, so the substance P producing neurons within the intestine and everywhere else inside the body. We then injected those mice with this fusion toxin, so this GTD coupled to inactive diphtheria toxin. And when you inject that into wild type mice, it has no effect on the mice. But when you inject it into mice that have the receptor, it causes that really profound edematous response that we associate with C. diff. And similarly, as a control, we actually injected basically a form of this GTD which can no longer interact with those GTPases, and that form no longer causes this pathological effect. We wanted to understand which exact neurons might be contributing to this kind of effect. So a really classic tool is to use resin for a toxin. It's basically super chilly. It's like so powerful it like knocks your socks off, and it knocks out some of the neurons that innervate the intestine, particularly the ones expressing TRPV1. When we did that, we observed that that decreased the ability of these toxins to cause substance P secretion. And similarly, that also decreases the ability of these toxins to cause a pathological effect within the intestine. We then wanted to go a step further by applying this in a kind of different system. Here we use NaV1.8 Cre. This is really selective for the neurons that come from outside your intestine, so there's extrinsic fibers. And when we delivered the toxin selectively into those neurons, so here using a germline cross, so the Cre is crossed directly to the flux DTR. In this case, we observed a really profound effect where we see really strong edema, really strong infiltration of immune cells just by intoxicating the neurons. So in summary, this kind of really revealed a really specific process of this toxin. So this intoxication of this kind of neurovascular apparatus, we think really drives this infection mechanism. So the TCV will enter into the tissue and then bind to particular cells and intoxicate them with high efficiency. So cells like the neurons and the pericytes from the intestine. And then this kind of coalescing response is really what drives those profound pathological changes. We then wanted to sort of understand what the effect of this was on the infection itself. So one of the things we observed is actually if you knock out these neuropeptides, it actually decreases the colonization performance of C. difficile itself. So it decreases its ability to efficiently colonize. This is actually reliant on toxins. So if you don't have the toxins, you no longer have a colonization advantage. So it decreases your ability to colonize. And that's actually dependent on neurogenic inflammation. And this is true either if you knock out substance P or if you knock out CGRP. Luckily for us, there's actually loads of really cool drugs that target these pathways, largely because they're sort of old failed pain targets. So these old failed pain drugs were very useful for us. When we pre-treated mice with these, it decreased the ability of the bug to colonize those mice, subsequently decreasing the burden of the infection. And this was true even in really hypervirulent strains. So strains like this one from the UK in the 2000s that sort of infected hundreds of people. And this is true either if we use small molecules or indeed if we use antibodies that can sort of neutralize CGRP itself. So receptor antagonists or antibodies targeting the neuropeptides very directly. This is really interesting, and some of my clinicians often will say, like, "Where's the meat? Like, how can this help my patients?" And in this case, what we actually found is that if we infected mice with a hypervirulent strain, so in this strain, sort of 80% of the mice would normally die, we observed really strong protective effects. So it protected about 80% of the mice in each case. And that was mirrored by reductions in the pathological effect within tissue. And what that really revealed is sort of this enhanced complexity of these mucosal interactions at these neurovascular surfaces. And I was kind of heartened. There's been many studies since this that have sort of extended upon these findings, showing how this can occur with different sets of neurons. So sympathetic neurons also contribute to this process. And indeed, other studies have shown how non-frizzled targeting variants of C. difficile also target neurons through other different pathways. So it seems to be a pathway that's very conserved between different forms of C. difficile. And lastly, in some sort of unpublished data, actually from the University of Pittsburgh, we actually studied patients who were taking migraine medication who also had inflammatory bowel disease. So these patients have a really high incidence of C. difficile. So we followed a cohort of patients who were being treated with either triptans or these molecules that block neurogenic inflammation, so these anti-CGRP therapeutics. And what we actually observed is in these patients, it dramatically reduces the incidence of C. difficile if they're currently taking these antibodies that block neurogenic inflammation. So lastly, I'd really like to thank my mentor for sort of doing this study with me. He's a fantastic mentor who taught me a lot about science and how to run a really good lab. And also, obviously, our collaborating labs, particularly Meenakshi Rao, a really talented gastroenterologist, and obviously Melissa Musa. My lab still works on all of these topics. We're super interested in understanding how pericytes contribute to vascular neuroimmune interactions. And we continue to study all kinds of toxins to understand exactly how specific targeting within tissue influences their pathogenic processes. And with that, like any questions?